Hi, everyone. We're going to wait just a couple of minutes to allow folks to join the session. While we wait, feel free to use the chat to say hello to your fellow attendees and let us know where you're from.
No, that's not a surprise. Baltimore, how about that? Welcome, Baltimore. Wisconsin, Chicago. We'll be there in a few months. Montana. Wow. Paris.
A lot of activity in the chat. I love it. Okay, why don't we get started? Hello, everyone. Thank you for joining the Marfan Foundation and its divisions for our webinar titled Variants of Uncertain Significance. I'm Lauren May, Senior VP of Research at the Marfan Foundation. I'm joined by my colleague, Stephanie Amdur-Clark, who's the director of our Help and Resource Center. Before we kick off our presentation, Michael Weimer, the Marfan Foundation's president and CEO, would like to say a few words. Michael?
Thanks very much, Lauren. I just wanted to come by and say hello and thank everybody for participating. I've been watching the number of signees go up by day. I think we had something like 200 in the first hour or so when we posted this, but as of this morning, there were over 400 folks that are either going to be with us today or will have an opportunity to see this in the future. Also wanted to thank, Dr. Loeys for, again, doing a seminar with us. He's an amazing member of our professional advisory board. He's kind of tied to Loeys-Dietz as well. And we're just wrapping up Loeys-Dietz month here at the foundation. But as you probably know, we also serve, Marfan, VEDS, and most recently Stickler syndrome. And if you're interested in a global virtual conference, we'll be global and we will be virtual this coming June. The dates are June 15 to June 18, and you'll be able to sign up on our website, hopefully in the not too distant future. So thank you for taking time out of your Saturday, especially those of you that are late at night, like Dr. Loeys is. And so with that, I'll turn it back to Lauren. Have a great time.
Thank you, Michael. For this webinar, we will use Zoom's Q&A functionality to field any questions you may have about today's presentation. Please select the Q&A tab on your screen to submit your questions anytime during the webinar, and we'll cover as many as possible at the end of the session. Also, this webinar is being recorded and will be posted to the Marfan Foundation's YouTube channel in the next several days. It is my pleasure to introduce our speaker today. Bart Loeys is Professor of Medical Genetics at the Center for Medical Genetics of the Antwerp University Hospital in Belgium and in the Department of Human Genetics in the Radboud University Medical Center in Nijmegen, the Netherlands. He trained as a pediatrician in Ghent, Belgium, and as a clinical geneticist in the McKusick-Nathans Institute for Genetic Medicine at the Johns Hopkins University Hospital in Baltimore, Maryland, USA. Dr. Loeys' special interests include connective tissue disorders, specifically heritable aortic aneurysm syndromes, and cardiogenomics. Dr. Loeys is also a member of the Foundation's Professional Advisory Board. Dr. Loeys' variants of uncertain significance are always a popular topic, and you're always a popular speaker. And if there's any doubt about that, Michael mentioned we had about 250 people register for this webinar within the first 48 hours of announcing it. Not bad. Thank you for being with us today to tackle this. The floor is yours.
Thank you, Lauren and Michael, for the very kind introduction. I think I'm sharing my slides now. Is that right?
Looks perfect, yes.
Okay, thank you. So it's my pleasure to talk tonight for me, the afternoon for most of you, about quite a challenging topic, which is variants of uncertain significance. which in the medical world are usually abbreviated as VUSs. So what a VUS result means for you and your family will be the ultimate goal of this presentation. So I divided this talk in 5 different questions. First, we'll briefly go over what is the known genetic causes of thoracic aortic aneurysm and dissection. Then we will talk a little bit about the details of DNA and how we examine DNA, because that's important to understand what a genetic variant is and how we classify them. And then we'll give specific attention to what we call class 4 and 5 variants and class 3 variants, which are the VUSs. And then finally, I will give some glimpse into the future of what will be the future of the class 3 variants. So let's start briefly with an introduction on the known genetic causes of thoracic aortic aneurysm and dissection. When you look at the broad group of aortic aneurysms that happen in the chest or the thorax, many of them do have a suspected genetic contribution. And we usually divide them in 2 major groups, being the ones that happen only in one individual, which we call sporadic cases, and they account for about 80% of all patients with thoracic aortic aneurysm and dissection. And on the other hand, we distinguish familial cases, which accounts for about 20%. So 1 out of 5 patients that do present with thoracic aortic aneurysm or dissection do have a positive family history. And within this group of familial cases or potentially hereditary cases, we can distinguish what we call syndromic forms, such as the Marfan syndrome, the Loeys-Dietz syndrome, vascular VEDS, arterial tortuosity. It's a long list. And what is characteristic for these is that in addition to the aneurysm, they most of the time have involvement of other organ systems, such as the eyes and skeleton. And this is in contrast to what we call the non-syndromic forms or familial thoracic aortic aneurysm and dissections, which only have an involvement of the aorta and don't present with other organ system involvement. Over the last decades, there has been tremendous growth in the number of genes that can underlie these conditions. In our center, we currently test routinely for about 48, but there might be different numbers depending on the genetic center and the choice of the genes that are involved. So if you look at those genes, there is many for which there is quite strong evidence that they cause aortic aneurysm. For others, it might be a little bit weaker. And a consequence of having so many genes is that we are able to identify causal or pathogenic variants in quite a lot of patients, but also the number of variants of uncertain significance increases. And I'll explain later in this presentation why that is. So testing more genes gives us more outcome, but probably also some additional degree of uncertainty. And that's not always easy to understand and to cope with. So overall, you can distinguish 3 main groups of genes that make the genetic architecture of thoracic aortic aneurysm and dissection. On the right hand, in the blue circle, you see genes that encode for proteins that are present in the extracellular matrix. And the most well-known examples are fibrillin-1, that's causing Marfan syndrome, or collagen type 3 that causes vascular Ehlers-Danlos syndrome. So this extracellular matrix gives some kind of structural rigidity to the aortic wall, and either components of that matrix or enzymes that modify that matrix can be involved in a cause of TAAD. On the left side, you see some genes that are important for the vascular smooth muscle cell contractile apparatus. In the aortic wall, there is a cell type called vascular smooth muscle cells that allows some kind of elasticity when the heart pumps against the aorta. When these genes or the variants in those genes lead to altered protein function of the vascular smooth muscle cell, they usually cause what we call the non-syndromic aortopathy. So only the aorta is involved, no other organ systems. And then at the bottom in green, you can see some genes that are involved in a signaling pathway called TGF-beta signaling. And those genes are linked to the Loeys-Dietz syndrome. TGF-beta signaling is an important signaling pathway that communicates what's happening around the cell in the extracellular matrix and gives signals back to the cell to tell the cell what it should produce in the aortic wall. Why is it important to look at all these genes, and can we not just focus on a couple of them? Well, the reason for that is that even in thoracic aortic aneurysm, one gene can sometimes present itself with more syndromic or less syndromic features, and one disease, for example, Loeys-Dietz syndrome, can be caused by multiple genes. So there is an increasing tendency everywhere in the world, genetic testing centers, to bring all these genes together and to analyze them as a set of genes that could be causal for thoracic aortic aneurysm. So how can we investigate that? Well, to better understand that, I first have to explain you a little bit what is DNA and how do we examine DNA. So here you can see a cell on the upper left corner. In the cell, there is a nucleus, and in that nucleus, we have chromosomes. And the chromosomes are the carriers of our DNA. And DNA, as you can see, is a double helix strand, which is composed of 4 letters, cytosine, guanine, adenine, and thymine. And we call these bases or nucleotides. And a sequence of such a number of nucleotides composes a gene. And a gene is nothing more than a series of letters following each other. But there is a coding system in it that allows it to be transcribed and translated into a protein. And a protein is composed of building blocks, and these building blocks form the protein. In order to understand that a little bit better, I like to make this comparison. So you can really compare our DNA to the English language. And the DNA, there is only 4 letters, bases or nucleotides, A, G, C, and T, whereas we know that in the English language, we have 26 letters. A combination of 3 of those bases or nucleotides we call a codon, and you can compare that to a word in the English language. So a gene would then be a sentence or a paragraph that is composed of different words, and a gene is composed of different codons. The genes are packed together in chromosomes. So one chromosome contains many genes. So that could be, in our comparison, a book. And when we talk about the genome, it's really the whole encyclopedia. So within our cell, each nucleus has 46 chromosomes, so 46 books, and they make up the whole genome or encyclopedia. Throughout this talk, you will also hear about some other word, which is called exome. And the exome is really containing all the important sentences. So the important sentences are the genes which contain the information for all the proteins that we need in our body to make the different organ systems and to make the body function. So when you try to remember this analogy, it will be easier to understand what genetic variation means. So over the last decade, or even 2 decades almost now, let's say starting in the 2010, genetics has really undergone a huge revolution, and that revolution is characterized by 2 major things. First of all, technology has allowed us to decode the complete human genome. And that's because machines were developed that were able to do that job. But not only the technology advanced, also the speed and cost has evolved over time. So in the past, when we had a suspicion that a patient had a specific disease, we could analyze only one gene at a time. So for example, if we suspected Marfan syndrome, we would first start with fibrillin 1. But maybe not every Marfan patient does really have Marfan syndrome, and it could be a condition that looks like Marfan syndrome, for example, Loeys-Dietz syndrome, and then we needed to look at other genes. So in that time period, it took a lot of time, and it was also quite expensive to identify those variants. With this new generation of sequencing machines, we can analyze not only many genes, but also many individuals at the same time, get a genetic result very rapidly, or more rapidly than in the past, and at a relative low cost. To give you an idea, making the code of the first genome costed about $3 billion US. 2 weeks ago, one of the sequencing companies announced that they would be able to do a whole genome for less than $100 US. So just to give you an idea of the huge magnitude of change of speed, the extent of genetic testing, and the lower cost of genetic testing. So we moved from one gene at a time to what we called initially panel gene testing. It would mean that from the DNA, we would select a limited set of genes, in this case, all the known TAAD genes. We would look for the code of a couple of thousands of letters, nucleotides, and that would be mostly covering the coding region. At the time, that was considered the best way because you would avoid incidental findings because you only look at genes that are important. But as I told you, technology is really evolving, and most labs now are shifting to what they call whole exome sequencing. So it means that we not only have the code of the genes that are involved in thoracic aortic aneurysms, we do have the code of all 21,000 genes, which is about 3 million nucleotides. And from that data set, the labs then select based on which patients they are testing. In our case, again, it would be patients that are at risk for aortic aneurysm dissection, and we analyze those sets. In a second phase, you could look beyond that set and look at coding information of other genes. But that brings with it the risk of incidental findings because you might find things that you're not really looking for and are not really related to the aneurysm cause. So that should be done with caution and only at a specific indication. And we're not done yet because, as I told you, prices are dropping and technology is continuing to evolve. And my prediction would be that quite soon we will also be moving to what we call whole genome sequencing. It means that we're not only looking at the coding information that's in the genes, but we will go for the 3 billion nucleotides, the whole genome, and look both at coding and non-coding regions. Technically, that's feasible. We can do it fast. The problem is the interpretation of the variation, especially when you go into the non-coding region. So the information that's hidden in the genome that does not lead to a protein, but could influence the amount of protein that's produced, for example, is more difficult to interpret. And again, here we have a risk of incidental findings, things that we wouldn't look for that we would identify. So if you want to use another analogy, as I told you, the DNA is hidden in the chromosomes in the nucleus of the cell, and it's really comparable to a cooking book. It has all the recipes to make many different recipes. All the ingredients could be compared to the genes or some of the codons. And if that information is right, you will get a nice cake. But if there is some mistakes in your recipe and some of the ingredients are not made in the right amount, or the quality of the ingredients is not good, then your cake will fail. And that's exactly what's happening in the human body. The genetic code can tolerate some variation, but at some specific spots, if you have a mistake in one of these letters, it will lead to a human genetic disease. So to translate that again to the aorta and the thoracic aortic aneurysm and dissection, at the top you can see the DNA. On the left side, you see a normal protein that's produced that gives a normal sized aorta. However, when there is an error in the DNA code, so when there is some variation, and we'll go further into detail later on, you can get either a bad protein or you can get no protein at all. And that will change, in this case, the rigidity of the aortic wall and lead to dilatation or aneurysm and dissection later on. So now we know what DNA is, how DNA can cause a disease. We will talk a little bit more about what genetic variants are and how we can classify them. So do we have variation in our genome? Yes, of course. And we do have quite a lot of variation. I told you that one genome is about 3 billion nucleotides, so 3 billion letters. That's a tree with 9 zeros behind it. And we do know that every person has on average about 4 million variations. I won't go into all the details, but it means that there is one genetic variant every 1,000 nucleotides in our genome. So if we're doing a genetic test and we're trying to find the right variant that explains the disease in our patient, for example, here that has an aneurysm, it's really looking for a needle in a haystack. And so we need to be able to distinguish among all this variation, which is the important variation. Because, and this is maybe the most challenging part of the presentation, not every genetic variant is harmful. And I'll explain that with 3 examples. So here you can see a piece of DNA code. So the letters are on top, what we call the nucleotides. You can see there is 4 of them, A, G, C, and T. Every 3 letters make one codon that has the information for a building block of the protein. So here we produce a protein. So now we can change letters. For example, if this A is transformed into a T, it will cause a variant at the genomic level, but it does not change the code. So the GTA message is the same as the GTT message. So there is some redundancy that nature has built in. So this will not lead to an altered protein, and as such will not be harmful. If you change a letter in another position, for example, if we change this C to an A, then there will be a change also in the code, and there will be another building block built into the protein, and you get altered protein quality. And this can be potentially harmful. We are not sure, but it could be. Lastly, another example, and I'm not discussing all possible genetic variations, I'm just giving you some general idea of what the consequences are. In this case, there will be a change of this C to a G, and this leads to what we call a premature stop of the protein. It means that the protein will not be produced in the right amount. So we get altered protein quantity. And that's very likely to be harmful. So when we do genetic testing and we find all these possible variation, and as I told you, there's a lot of variation, we have to try to sift through it and determine which ones will be the ones that are important. Obviously, the ones on the left, we will not bother about because they don't do anything. The ones in the middle, we will think about and we'll have to study and see what they do. The one on the right might be the one that is very likely to be harmful. So it's like the stone of Rosetta. We always find genetic variants, but what do they mean? Are they helpful or not? Are they disease-causing, or are they just a variation of normal? And there is also, over the last decades, a little bit of an evolution of the terms that we use to describe this variation. In the old genetic world, let's say more than 10 years ago, 20 years ago, we were talking about what we call mutations, which usually were considered as very rare and disease-causing. And on the other hand, variation that was called polymorphisms, which are common and considered benign. However, in the new genetic world, we only keep talking about genetic variants. And these variants are now classified into 5 different groups. And that will be the main focus of the presentation. So a class 1 and 2 are the less harmful or not harmful, benign, likely benign. The class 3 are the ones that are in the middle and quite uncertain. And class 4 and 5 are likely pathogenic or pathogenic. So these are more likely to be harmful. So that was introduced a couple of years ago by the American College of Medical Genetics. So to give all the genetic clinics the same language, they developed guidelines and standards on how you should call a specific genetic variant. This is a very complex process. I won't explain this table, but this is just a general overview of all kinds of criteria that are used to try to classify these variants to find arguments why something would be harmful or not. Even for us as geneticists and molecular geneticists, this is a very complex system, and it would lead us way too far to explain it in detail. Moreover, it's in the near future probably also going to change a little bit. So I'm just going to highlight which type of data that we use to try to come to a conclusion. And I will explain to you how these data are interpreted and how they give less or more weight to the pathogenicity of a variant. So there will be 6 items. The first one is population data. What does it mean? When we find a variant in a patient and we find it suspicious, we will always look how often this variant is present in healthy control databases. Due to the advancement of genetic technology, we have now access to a large control database. It's a database where the genomes of about 1.6 million copies of each gene are represented. It's called GNOMAD. And so the idea is that the rarer a genetic variant is in this large control population, for example, if you see it only once in the 1.6 million gene copies that are in the database, it is more likely to be pathogenic. As you can see on the left, this is also somewhat population specific because there might be some natural variation that is present in specific populations, depending what your region of origin is. So that can also be taken into account. If you know the ethnic origin of the patient, you have to match it with the appropriate control population. So just remember, the more rare the variant is, the more likely it is to be pathogenic, because if it's not present in the controls, it's more likely to be disease-causing. A second step that we take is that we look at the protein that is predicted by the genetic code, and we do two things. First of all, we look throughout evolution if a specific building block is highly conserved. And the idea behind that is the more that specific building block is conserved, the more likely it will change into something pathogenic. Because evolution tries to keep things that are good. So you don't want to change good proteins, and these have a highly conserved genetic code, a highly conserved protein code. So, for example, here you can see part of a code, and we look throughout evolution, mouse, chimp, gorilla, rat, cow, cat, dog, chicken, zebrafish. And you can see that at the same position, there's always the same building block in the protein. So if you change something to that, it's very likely that it will cause disease. Secondly, we also look at the details of the composition of the protein. And some building blocks are more important than others. And I'm showing here it's for fibrillin 1. Fibrillin 1 has these yellow building blocks which make bridges with each other. If you disturb such a building block, then that bridge will also be disturbed and it will change the structure of the protein. So variants that impact critical protein domains are also more likely to be pathogenic. So that requires some exercise to look in databases whether that's the case or not. A third, and that's a very important one because it can give really a lot of evidence for pathogenicity, is to do a functional study. It means that in the laboratory, we will create an experiment where we can really study the effect of the genetic variation on the protein. Unfortunately, for many of the TAAD genes, we don't have such a good functional assay. I'm showing here two examples that we are working on in our lab. For example, by glycan, you can see here normal protein that's produced, and those patients don't produce any biglycan. So that's a very strong evidence that this protein is really missing and that your genetic cause is leading to a deficiency of the protein. Similarly, for SMAD3, we're trying to develop an assay where we can convert the activity of SMAD3 into the activity of luciferase. So it will produce some signal that we can measure, and then we can see whether that's normal or not. So a functional test can demonstrate in an experiment in the lab that a genetic variant leads to an altered protein function or lack of the protein and suggests some biological mechanism. So this is a very strong but very hard to get argument. And for example, for fibrillin 1, we don't have such a test. For the TGF-beta receptor 1 and 2, we don't have such a good test yet. So that would help to classify VUSs if we can find more better functional tests. But that's research that's ongoing, and that will evolve over time. A fourth factor that we look at is whether we can find the genetic variant in another affected individual in the scientific literature or international databases. And there's many databases now that list variants in patients that have been seen before. And so if you identify the exact same variant as a variant that has been seen in another patient with a specific disease, that adds very strong evidence that this is not going to be something benign, but leading to disease. Then number 5, and that's also a quite difficult one. Not every gene that we know that leads to aortic aneurysm has the same validity. It means that the evidence that this gene is leading to aneurysm is not always so strong. And there is expert groups. For example, this was a paper that was published in 2018, where a lot of experts from the whole world came together, went through all the genes and gave some kind of evidence, being very strong, moderate, or limited evidence that this gene is really an aneurysm-causing gene or really is a disease-causing gene. So it's very important to match the clinical features, so what is present in the patient, to the gene and see if there is a strong match between those two. I will give an example. If we do see a patient that has a lens dislocation and an aortic aneurysm, and we subsequently identify a variant in fibrillin 1, then it's very likely that those two explain each other, because there is only one gene that we know in the scientific world that can cause ectopia lentis and aortic aneurysm in the same patient, and that's FBN1. So if you find a nice match between what you expect to see as the phenotype and the gene variant, then it's again adding evidence for validity of that specific variant. Lastly, we can also look in the family, and that's something that we call a segregation analysis. In the first example here, I'm showing a young patient that unfortunately died due to an aortic dissection at young age, in which we identified a variant in TGF-beta receptor 2, which is one of the genes for Loeys-Dietz syndrome. And what we did is we checked in all the family members. Here we have the father, the mother, and a brother. They're all unaffected. They don't have aortic aneurysm. They don't have signs of Loeys-Dietz syndrome. And we've shown that that variant is absent in all of these. So it's something that we call de novo. It happens upon formation of the sperm or the egg from the mother. The whole genetic material has to be transcribed. Nature made a mistake, and that's a new mistake. So finding a de novo occurrence in an affected patient with unaffected parents not carrying the variants is a very strong argument for pathogenicity. Secondly, you can also do, in case there is a family history, segregation. And in this case, this is a family with non-syndromic aortic aneurysm and dissection. We found a variant in ACTA2. And what we try to do is we try to show that all the individuals that are colored here black, it means that they are affected, have the mutation. That's where the pluses are. And all the patients and individuals that do not have the aortic aneurysm and dissection, they have a minus, which means they don't have the variant. So showing that a potential variant, potential pathogenic variant, is present in all affected individuals and absent in all unaffected individuals adds again strong evidence that this variant is going to be pathogenic. So determining this likelihood of pathogenicity is really a complex exercise that is taking place in the laboratory, but also needs a clinical input. It needs to match the clinical findings with the family history, potential segregation studies that show de novo occurrence. And then you can bring all that information together and try to come to a conclusion and put it in one of the 5 categories. So for example, it could be that what we have identified is common or even very common in the healthy population. So for example, we see it in more than 500, 600 people in the control database, well, that means that it's very unlikely to be disease-causing because we see it too often in healthy people. So that will most likely lead to a class 1 or class 2 benign or likely benign. A lot of variants end in this middle category. For example, you can have a variant that's very rare, so you don't see it in a lot of control patients. For example, only once out of 1.5 million copies of the gene in our control database, but we don't have any good functional studies. And then very often it will end as a class 3 or a VUS. When can you get to a class 4? For example, when the variant is absent in the control, so extremely rare, and because it's not present in the 1.6 million copies, and we do have functional studies. If you can combine those two, it's very strong evidence, and it will be likely pathogenic. Pathogenic will be often when it has been seen already before in affected individuals, and there is a well-established effect on the protein function and the biological mechanism. So that's how this classification occurs. And to put it in statistics, and that's the framework that's behind this whole classification system, you can put a number of likelihood of pathogenicity on the different classes. So for the class 1 and 2, it's less than 10%. For the class 4 and 5, it's more than 90 or even more than 95% certain that this variant is indeed pathogenic. And the problem is the large middle group, which is between 10 and 90% the variants of uncertain significance. I think it's important to realize that this conclusion can only be made also with a good clinical evaluation. And it's also important to know that this is a dynamic process. It evolves over time. It's not fixed. A variant of unknown significance can over time evolve to a likely pathogenic or a likely benign. So how do we act on this, and what does it mean? The first step is, of course, what is the laboratory reporting? In general, you should know that class 1 and 2 will not even appear in your reports because they are not important. The class 4 and 5, of course, will always be in the report. The class 3, it's a little bit dependent on the laboratory, what decision they take to report it or not. So what do we do with this classification? So first of all, if you have a class 4 or 5, why is that important? Well, I think the first thing is that it resolves a diagnostic odyssey. People have been looking a long time for an explanation for what's happening in the family. A class 4 or 5 variant gives that explanation. It tells us also a little bit about the natural history. It means that we can start comparing the patient to other patients that have genetic variants, same type of genetic variants in the same gene. For example, in the aortic aneurysm world, it more and more starts to guide what our management will be. Do we need to look at other organ systems or not? There is also evidence more and more that it also guides the thresholds that, for example, we use to do surgery when the aorta dilatation becomes too big. So it's really important to try to get to this class 4 and 5. Another consequence is that in prenatal diagnostics or in vitro fertilization with pre-implantation diagnosis, that will only be done when the variant is classified as a class 4 or 5, because the predictive value is big enough, strong enough to say or to know that this variant will be disease-causing. So we can also do predictive testing. That's something that we call cascade testing. It means that in the first-degree relatives, so children, brothers, sister, parents, we can look for the presence of that variant. And based on the absence or the presence, we can reassure them, or we can start additional screening. For the class 3, that's a little bit more difficult, and there's quite some debate on how to use these variants. And I will discuss that in a little bit more detail. So this class 3 variant is a variant that's a change in a DNA for which we don't know whether it's harmful or not. It's like finding a spelling error in a word, and sometimes it will change the meaning, sometimes it doesn't, and sometimes we just don't know yet. So it's very important that a class 3 variant does not confirm a diagnosis. It's a possibility, but definitely not a final answer. Secondly, it also does not mean that you and your family members will develop the disease if they have this variant, because we don't know what the significance is. And as such, it should not be used alone to make major medical decisions. So you cannot just on a class 3 variant do predictive testing in the family because it doesn't tell you enough. So in summary, that's how the significance of these variants is evolving. The class 5s, you can base clinical decisions. Most of the time, this is also the case with likely pathogenic, but you cannot do that with a variant of unknown significance. So what is the usefulness then of this class 3 variants? Well, I will explain you a little bit how we try in the medical world, in the genetics world, to learn a little bit more about the significance of these variants, how we try to downplay them either to class 2 or to upgrade them to class 4s. And that's what I like to use the terms of cold and hot VUSs. A hot VUS is a VUS that is more leaning towards a class 4, whereas a cold VUS is probably not harmful and will be more likely likely benign. So we could say that a cold class 3 is more like a 2.5, and a hot class 3 is more like a 3.5, going towards 4. And this decision is made with the team, with physicians taking care of you in collaboration with the lab. How does it change our management? Well, when it's a cold class 3, we will do only a clinical screening of the first-degree relatives. In this case, it would mean that we would recommend to do an aortic screening by echo, CT, MRA, and we will not use genetics in the first line, but we will only do genetics if there is an abnormal clinical screening, because if that person also has the variant, it adds evidence that this is indeed disease-causing. And on the long term, if we have enough individuals in that family, we might upgrade it. If it's considered more like a hot class 3, a very suspicious class 3, we will combine the clinical screening. So we will do the echoes, CTs to look at the aortas, and we will do a segregation. And we will base our interval of follow-up on the genetic testing results. So everybody will get follow-up, but the ones that do have this class 3 variant, maybe will get it a little bit more often. For example, you could say in a family member that does have the variant, but a normal echo at the beginning, we will repeat it once every year, every 2 years. If that individual does not have the variant, we will not stop the clinical screening. As I said, you should never go by the absence or presence of a class 3 variant by itself. We will say we'll see you back in 3 to 5 years. So that's how we try to learn more about these class 3 variants. Are they an important problem? I will just give you a real world example. We did a multi-center patient assessment of patients that presented only with dissection, so not aneurysm. And we tested the 23 most relevant genes, and this is how the result looks like. So somewhere between 10 and 15%, you will find a likely pathogenic variant, so class 4 or 5. We didn't find anything in about 65%. And in 26%, we found variants of uncertain significance. So it's not uncommon to find these variants. What's the difference if you look at the incidence of dissection? So that's the age at which those patients develop dissection. You can see that the patients with the class 4 and 5s, which are in red here, are suffering for aneurysm and dissection about 10 years earlier than the ones that did not have a variant or were negative. And at every age, there was about a twofold higher risk of aneurysm. But you can also see that the green line with the VUSs does not completely overlap with the negative patients. So there might be some residual signal there that we might need to try to catch up. So I think over the years, class 3 variants could evolve towards class 2 variants, or they will emerge as class 4, depending on the follow-up that we can do in the family. To end, I would like to show you how these variants might work together also to lead to disease. So if you have a class 4 or class 5 variant, that might in itself be enough to cause your aortic aneurysm. Maybe in some rare instances, you need a small additional factor to tip you over. For example, having some hypertension on a class 4 or 5 will cause your aneurysm. There might be also aneurysms that are caused by variation that's quite common, and a lot of these variations together might also cause a problem. And for the class 3s here in green, it might be that maybe if you have 1 or 2 of those class 3s, that you need an additional factor. For example, the hypertension, that might also be enough to reach the threshold of an aneurysm. So with this graph, I'm showing you that a more comprehensive evaluation of the DNA of an individual over time might learn us also about what we call oligogenic and polygenic, where it's not just one variant, but a combination of variants. And we should never forget this. We have 21,000 genes. So this single variant that we identify as a potential variant is acting within a background of a lot of other genes, and this should all be taken into account. So how do we deal with the clinical channels of VUS? I fully understand that this leads to confusion and anxiety, and this uncertainty can be very frustrating, and I think this is totally normal feeling. However, I think it's also important to realize that something found does not always mean something is wrong. It can evolve over time, and variants can be reclassified as harmless to class 1 and 2 or disease-causing class 4 and 5. So how can you help or how can you discuss this with your treating physician? I think it's important to see whether the additional value of family testing is there. As I said, it's very important to put all the clinical data, what the features are that are present back against the genetic data, and important to follow up with your clinical genetics team to do re-evaluation over time, because it's not because we don't know now that we might not know it in the future. And I would like to thank my team for supporting us and you for your attention. And I'm happy to take some questions.
Dr. Loeys, thank you so much for sharing your time today to discuss a topic that means a lot to our community. Members of our audience have already submitted many questions, and we will go through those now. For someone with a VUS for one of the LDS genes, is it necessary to monitor them regularly with imaging to look for aortic aneurysms or aneurysms in other locations? And not just maybe for LDS, but other conditions as well.
Yes. So as I explained, we do offer monitoring. So where a VUS can make a difference is maybe the timing or the interval between the exercise between the exams. So in general, we would follow up patients with likely pathogenic variants maybe every year. For a patient with a VUS, that might be spread out a little bit in the time, and you might only need to do it every 2 or 3 years because the effect is less strong or not so certain. And of course, that also depends on the first imaging study that was done. I think it is important in the families where there is a VUS that not only the family members that do have the VUS are followed up, but also the family members that don't have the VUS, because we are not certain, and it's safer to follow up these individuals also, maybe with a little bit longer interval, let's say every 3 to 5 years.
Okay, great, thank you. Do genetics teams automatically revisit patients' genetic data as new information becomes available, or is that something that patients have to advocate for?
Well, it's always important to be your own advocate. That's good. But in general, if a variant is upgraded from a class 3 to, let's say, class 4, the lab will retrospectively look whether there is other patients that have the same variant and whether they should be alerted. And then a letter will go to the referring physician. If it's downgraded, usually that effort is not done. So it's more when it goes into pathogenicity that there is a practice to contact the referring physicians again.
Okay. How are individuals with multiple TAAD-associated VUSs evaluated? And how common is that it for clinical genetic testing, excuse me, to return multiple TAAD-associated VUSs?
I would say it's not so uncommon. Of course, it depends a little bit on how stringent the lab is on what they decide to report. There is labs that have a very strong policy, and they will only report maybe the stronger VUSs, like the 3.5 that I talked about. Others are putting the threshold a little bit lower, and they might report the 2.5s also. And then you will end up with more variants. I think, again, doing the segregation study in the family, always combined with the clinical testing, so the echo, CT, imaging, physical exam, might shed a light on which variants are important and which aren't, and which individuals need follow-up and which don't.
Okay.
But that's really an individual decision.
Understood. Thank you. We have a few questions about folks who have a VUS. How can they best contribute to research or to advancing the understanding of that VUS to something more concrete? Are there places where they can contribute their genetic data or family history?
Yeah. So there is a quite large international effort, which is called ClinVar. And a lot of diagnostic laboratories contribute their variants into those databases, which allows other labs to see then if somewhere else in the world the same variant has been identified in the same gene, and then there could be exchange of information at the laboratory level. And then with permission of the patients, there could be even be exchange of the clinical information. I think there is growing importance of registries. For example, in Europe, we have the European reference networks, which try to catch all that genetic variation and align it with good clinical information, because, as I said, it's always the combination of the two, not just the lab, but also the clinical contribution on what was the exact phenotype, how was it in the family, was it de novo or not, that helps to classify. So you could ask your physicians to submit your variants into those databases, and then get feedback when there is matches that show the exact same variant.
Okay. Thank you. Are there synergistic effects? Are synergistic effects evaluated? Excuse me, especially if the gene pathways overlap and may plausibly exacerbate a presentation?
Yeah, it's a very good question. It's very hard to test that because it's already difficult to have a functional test for one gene. Let's say it's extremely hard to test that for two. That can be done, but that will be most of the time will be in a research setting and not in a clinical diagnostic service. So it's really very experimental and very research-wise.
Okay. Two more questions because we are running out of time. This person was molecularly diagnosed with VEDS in 2013. Is there anything more that they could learn if they were to have genetic testing performed again now?
Specifically for vascular EDS? No, I think if the variant that was identified at that time was considered to be pathogenic, that most likely will still be the case. We do know a little bit more about genotype-phenotype correlation in vascular EDS, so the type of variant might be relevant to be discussed again because the natural history is somewhat different for different types of variants. My prediction will be that in the future, that will probably change. And as I said, a genetic variant acts in the context of 21,000 other genes. And so it's highly likely that some variation in other genes that we are investigating now will modify the outcome of the primary variant. So for example, in this case, if that patient has a pathogenic variant in COL3, we do know from observation that sometimes individuals with the exact same variant have not the exact same phenotype, one being severely affected and another one not being so severely affected. And one of the ideas, one of the hypotheses is that variation in the 20,999 other genes do influence the expression of this variant. And that's research that's going on. And with gathering more genomic data, we will be able to make better predictions of combinations of genes also. But for that, we need extremely large data sets and good clinical phenotyping. But that's definitely something that's going to come.
Okay, great. Last question. Do you know of any ongoing research related to VUSs for fibrillin 2?
That's a good question. Fibrillin 2 is a very difficult gene because it has indeed many VUSs. Myself, I'm not convinced that fibrillin 2 is a bona fide thoracic aortic aneurysm gene. There is some reports of patients with aneurysms that also have other features that are associated with this condition, being the Beal syndrome or congenital contractile arencapacity. So I think it's not completely impossible. But one of the major concerns is that fibrillin 2 is expressed at very low levels in the postnatal period. So that means that during the embryonic development, when you're a baby in the womb, fibrillin 2 has an important role. But after birth, the role of fibrillin 2 is taken over by fibrillin 1. And that's why we have Marfan syndrome and thoracic aortic aneurysm. From a biological perspective, it's a little bit hard to explain that a protein that's not highly expressed in the aorta would still be capable to cause an aneurysm. So that's why I'm a little bit doubting whether it by itself could be a cause. What I could imagine is that it is a cause in the context of a combination with other things. So kind of maybe a risk factor where I showed at the end where you have 2 or 3 variants that come together. And at this time, we might not be able to identify those other variants. But I'm hoping that with more genetic analysis, it will become clear what brings maybe these variants in fibrillin 2 above the threshold and leading to aortic aneurysm. But it's a very challenging question. I'm not sure I know the full answer, but these are my thoughts at this time.
Okay, we appreciate that. Many thanks to those who shared their questions, and obviously to Dr. Loeys for addressing them. Staff in the Marfan Foundation's Help and Resource Center can assist with questions about your specific situation. So if we didn't get to it, a question that you asked, please feel free to direct those to marfan.org/ask, and someone will be in touch. Let me just share my screen one more time.
You can forward it also to me then later on.
Wonderful. Thank you. Thank you, Dr. Loeys. Okay. Before you go, please take a moment to share your feedback on this webinar by visiting marfan.org/survey or scanning the QR code on your screen. Thank you so much for joining us today. Take care.